Good afternoon, students. I hope you are all fine. Welcome back to the biology class. So, today we will be starting with the post fertilization changes because yesterday we completed up to double fertilization. So, we'll take the next topic now, that is post fertilization changes. But let me tell you one thing before I start, that I am going to test you after the completion of the chapter. So please be prepared for it. Okay. You should do your work regularly and daily. So please be attentive and be prepared. Now let's come back to the topic and start with today's topic. We start from the post fertilization changes. Now after fertilization comes the development. Now what sort of development? There are certain changes in the flower first of all. Now what are these changes? The flower begins to lose its shine. The petals, stamens and style, they either fall off or they wither away. The calyx, however, may persist in some cases. For example, you see the top up in tomato, brinjal, that is the persistent calyx. Now the major events which take place during this is displayed on your screen, and we are going to take one by one all these events. So the first is the development of endosperm from diploid primary endosperm nucleus in the central cell of embryo sac. Second is the development of embryo from diploid zygote. Third is the development of seed from ovule, and last is development of fruit from ovary. Right? So have you seen all these four? Have you seen them? So now we'll take these one by one. The first one we'll start with, that is the development of endosperm from diploid primary endosperm nucleus in the central cell of embryo sac. Here we start with. So endosperm is the food-laden tissue formed during the development of angiospermous seed, which provides essential nutrients to the growing embryo and also the young seedling at the time of seed germination. However, in gymnosperms, the female gametophyte is known as endosperm. But in angiosperms, the endosperm develops from triploid, that is its ploidy is 3N. From the triploid primary endosperm nucleus, which is formed as a result of vegetative fertilization or triple fusion, or that is a fusion of one male gamete with the secondary nucleus of the central cell. We discussed this yesterday, if you recall. Now, based on the first and subsequent divisions of the primary endosperm nucleus, that is PEN, the endosperm is of three types, that is nuclear, cellular and halobial. We'll take these three one by one. The one displayed on your screens is the nuclear endosperm. Now what happens here in the nuclear endosperm, you can see this is the diagram of the nuclear endosperm, and in this type of endosperm development, the primary endosperm nucleus divides by repeated mitotic free nuclear divisions without the formation of walls, that is the cell walls. It results in the formation of large number of free nuclei in the central cell of the embryo sac. A big central vacuole develops in the embryo sac, pushing all the nuclei to the peripheral cytoplasm. The free nuclei come to lie between the embryo sac, cell wall and the vacuolar membrane of the central cell. The second phase begins with the cleavage of multinucleate peripheral cytoplasm, resulting in the centripetal formation of cell walls. Finally, all or most of the endosperm is converted into a cellular tissue. For example, coconut. In coconut, The endosperm is multicellular in the outer part and free nuclear in the center. Nuclear endosperm is the most common type of endosperm. Here you can see in the diagram also it is displayed that first the nuclei is dividing without cytokinesis and finally the cellular endosperm is formed. Now next we come down to the second type, that is the cellular endosperm. In cellular type of endosperm development, the first nuclear division of the primary endosperm nucleus is followed by the formation of either a longitudinal or transverse cell wall in the central cell. Subsequent nuclear divisions and wall formations result in the formation of a cellular type of endosperm, and examples are like petunia, datura, balsam. So here you see the cellular type of endosperm displayed on your screens, where you see the cytokinesis is being followed by the nuclear division. So now we come down to the third type, that is the halobial endosperm. It is displayed here on your screens, the halobial endosperm. In the halobial type of endosperm development, the primary endosperm nucleus moves to the chalazal end of the embryo sac. The first nuclear division of the primary endosperm nucleus is followed by the formation of the transverse wall, resulting in the formation of a small chalazal cell and a large micropylar cell. Generally, the chalazal cell does not divide further and functions as hostorium. Nucleus of the large micropylar cell divides by repeated free nuclear divisions, and further development takes place in the same way as the nuclear endosperm. Halobial type of endosperm development is prevalent in monocotyledons. Is it clear now? The MC here in the diagram means the micropylar cell, and the CC here in the diagram represents the chalazal cell. So you can see the presence of the and the position also of the zygote and the endosperm. Now we come to the second point, that is development of embryo from the diploid zygote. Now children, before I discuss this, let me tell you that this endosperm and the types of endosperm is a very important question from examination point of view, and a 3 marks question is directly comes for the 3 different types of endosperm development. And one more thing, let me tell you about the endosperms, that why the endosperm develops after the fertilization. As I told you earlier also, that nature is very conservative whereas the energy is concerned. So it does not want to waste its energy for the formation of the endosperm before fertilization. So if the fertilization is affected, only then there is need for the development of endosperm. That is why it develops after the fertilization. Is that clear? Okay, so now let's proceed with the second part, that is the development of embryo. Now as far as the development of embryo is concerned, it is broadly of the 2 different types. One is that is in the dicots, and the second is that in the monocots. So first we will take up the development of the embryo in the dicots. So it is displayed here, the various stages of the division. They are displayed on your screens, so please listen to me carefully. In a typically dicot plant, for example like in Capsella bursa pastoris, the type of embryo development is typical, right? So we will take this up. The zygote enlarges in size first, secretes its own cellulose cell wall, and then divides by a transverse division, resulting in the formation of 2 unequal cells. You can see in the diagram which is displayed on your screens. The larger basal cell is called suspensor cell, which lies towards the micropylar end of the embryo sac, And the small terminal cell is known as the embryonal cell, which lies towards the chalazial end of the embryo sac. The basal suspensor cell divides by a transverse division, and the terminal embryonal cell divides by the longitudinal division to form a tetrad. Out of the two cells formed from transverse division of the suspensor cell, the lowermost cell towards the micropylar end enlarges in size to form a vesicular basal cell. The other cell divides by a variable number of transverse divisions to form a long suspensor consisting of a row of 7 to 8 cells. Each of the two terminal cells of the tetrad stage of proembryo divide longitudinally now, forming 4 cells, which is known as the quadrant stage. Each of the 4 cells of the quadrant stage then divides by a transverse division to form 8 cells, octant stage. Each of the 8 cells of the octant stage then divides by periclinal division to form an outer protodermal cell and an inner cell. Consequently, the 8-celled octant now becomes 16-celled. The 8 external protodermal cells and 8 internal cells. Right? Okay. So then these 8 protodermal cells, they divide by anticlinal divisions to form the embryonic surface layer or epidermis. The 8 inner cells divide in different planes to form the ground meristem and procambium of cotyledons, hypocotyle and plumule. Now the suspensor cell situated at the terminal end of the suspensor, which is in contact with the globular proembryo, is known as hypophysis. You can see here in the diagram. The hypophysis divides by a transverse and longitudinal divisions to form 3 tiers of cells. The cells of the inner tier towards embryo forms the cortical initials of the radicle, that is the embryonic root. Here you can see. The cells of the middle and outer tier form the initials of root protoderm and the root cap. So all the cells derived from hypophysis give rise to root apical meristem. At this stage, the developing embryo is globular and shows radial symmetry. It is known as proembryo. The proembryo is transformed into embryo with the development of radicle, plumule and cotyledons. So the two cotyledons arise from the two ridges of tissues appeared at the distal region of the proembryo, so that the globular proembryo becomes heart-shaped embryo. Cotyledons grow rapidly, whereas a few cells between the bases of two cotyledons differentiate to form a dome-shaped plumule, which forms the future shoot apex. So this is how the complete development of the dicot takes place. You can see here these stages which are displayed on your screen. So here you can see the two cotyledons, you can see the plumule, the radicle, then the root cap, everything you can see in the last diagram, that is the K1. So here this is a completely developed dicot embryo. Now as far as the monocot embryo is concerned, we will take up now the monocot embryo. Here these are the different stages of the development of a monocot embryo. You can see them displayed on your screen. As you are all aware that the embryos of monocots, they possess only one cotyledon, right? Because the main difference, dicot, monocot. In dicot, it is two cotyledons, and in monocots, it is one cotyledon. So, in the early phases of the embryogenesis up to the formation of the globular proembryo, they are very similar in both the dicot as well as the monocots. But in monocots, about one half of the terminal cell derivatives, they function as the shoot apex, whereas the remainder of the cells, they show a rapid rate of cell division in the formation of single terminal cotyledon. And this cotyledon in the grass family, it is known by the name of scutellum. So, this scutellum is situated towards the lateral side of the embryonic axis. So, at the lower end, the embryonal axis has the radicle and the root cap enclosed in an undifferentiated sheet which is known as coleorhiza, and the portion of the embryonal axis above the level of attachment of scutellum is epicotyle, and this epicotyle has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure which is known as coleopetile. Here you can see in the different stages. So, now we come down to the next topic, that is development of seed. I hope up to here you don't have any problems, and if you have any problem, children, please text me. I will clear them. I will clear the doubts. So, next comes down the development of seed. This is the last topic of today's lecture. So, we have to take both the seeds, that is the dicot seed as well as the monocot seed. So, in angiosperms, the seed is the final product of sexual reproduction. It is often described as a fertilized ovule. Seeds are formed inside fruits. A seed typically consists of seed coats, cotyledon, and an embryo axis. The cotyledons of the embryo are simple structures, generally thick and swollen due to storage of food reserves. Mature seeds may be non-albuminous or they may be albuminous. Non-albuminous seeds have no residual endosperm as it is completely consumed during the embryo development, for example in pea or in groundnuts. Albuminous seeds retain a part of the endosperm as it is not completely used up during the embryo development, for example wheat, maize, barley, castor, sunflower. So, occasionally in some seeds such as black pepper and beet, remnants of nucellus are also persistent. Now, this residual persistent nucellus is known as perisperm. Remember the term, please. Integuments of ovules harden as tough protective seed coats. The micropyle remains as a small pore in the seed coat. This facilitates entry of oxygen and water into the seed during germination. As the seed matures, its water content is reduced and seeds become relatively dry. At this stage, they retain around 10 to 15% of moisture by mass. The general metabolic activity of the embryo slows down. The embryo may enter a state of inactivity called dormancy, or if favorable conditions are available, that is enough amount of moisture, oxygen and suitable temperature, then they germinate. Otherwise, they first enter the inactive stage, which is known as dormancy, or the seeds are known as dormant. As the ovules mature into seeds, the ovary develops into fruit. This is very important. The ovules, they mature into seeds, and the ovary, it develops into a fruit. So the transformation of ovules into seeds and ovary into fruit, they proceed simultaneously. Okay. So the wall of ovary develops into the wall of fruit, and this is known as pericarp. So now I've told you two terms, perisperm and pericarp. I hope you can tell me the difference between these two if you heard me properly and with concentration. And children, now we will discuss the fruit, formation of fruit and its types in tomorrow's lesson or tomorrow's class. Okay, so I hope this is clear to you. But the seeds, you can see both the monocot as well as the dicot seed. Here you can see in the dicot seed the presence of two cotyledons. Then you can see the presence of this plumule with the rudimentary leaves, the epicotyle, the hypocotyle, the radicle, the testa. All these things you can see in a dicot seed. And in a monocot seed, here you can see the presence of endosperm, which is filling up most of the part, and then above it is the aileron layer and the pericarp and the seed coat. Then and on the right side is present, here I'll enlarge it for you. Here is the scutellum, which is the cotyledon, then the coleoptile, I told this term before, the embryo, the radicle, the root cap and the coleorhiza. You can see all the position of all these things in the monocot seed. Here, I hope you can see it clearly. You can perform a small activity also at home. You can soak a few seeds in water. You can soak seeds of wheat, maize, peas, chickpea, anything which you have at home, any pulse or any grain which you have at home. Just soak it overnight and then take out the extra water and then allow it to sprout. When it starts sprouting, then split the seed and observe different parts of the embryo. You will be able to see all the different parts. I think you have all done this in your lower classes also, and you all eat the sprouted grains also. So sprouted grains are what you have to see actually in order to see the cotyledons and the presence of the radicle and the plumule. So you can perform it at home if you want to see it more properly. So children, now I wish you luck and best of studies till we meet tomorrow. So we'll meet tomorrow now at the same time for our next class. Thank you, children.